DNA and Cellular Adaptations, Section 2. Last time we looked at the structure of DNA, took a close look at what makes up DNA, the little building blocks called nucleotides. And we mentioned that DNA can be divided into sections called genes, and that each gene acts as the code to create a protein. Well, today we're going to take a step back and look at DNA as the smallest part of a group of structures. DNA, a single DNA molecule from beginning to end, creates a chromosome by wrapping around some proteins. And that chromosome can be found in a cell. If the cell is a eukaryote, the chromosomes will be found in the nucleus, which is a separate little section of the cell created by a membrane that wraps around the area where the chromosomes are stored. A chromosome is one molecule of DNA from start to finish, and different species, like human versus dog, have a different number of chromosomes. So we have different amounts of chromosomes between different species. Prokaryotic cells, like bacteria, have just one chromosome, and it is circular instead of linear, meaning a line. Eukaryotic cells have this linear structure that you see, and our chromosomes exist in pairs because we got half of our chromosomes from our mom and half of our chromosomes from our dad. And our chromosomes, because we are eukaryotic, are found in the nucleus. Every single body cell, like liver cell, heart cell, skin cell, in a single organism, like one person, one dog, one cat, would have identical copies of the organism's DNA. If I looked at one of your brain cells, I would find DNA identical to the DNA in one of your intestine cells. Each cell in your body has the same access to the same copy in their cell. Here we have a picture again of a cell and a single chromosome from that cell. And if you unwind the chromosome, you will find it's made of a molecule of DNA, which can be divided into sections called genes. Each gene would have hundreds of nucleotides as its building block. How many molecules of DNA would you find in each cell, every single cell, in the body of one of those species if that species has 8 chromosomes? Every single cell in that organism would have 8 chromosomes. So when we say a species has 8 chromosomes, like the fruit fly, which is the species that has 8 chromosomes, every cell in the fruit fly's body would have 8 chromosomes in the nucleus of its cells. Is the described cell, the 8 chromosomes, prokaryote or eukaryote? Well, remember, prokaryotes have just a single chromosome, just one. But eukaryotes have chromosomes that exist in pairs, so it's going to be an even number, like 8. Since this is more than one and it's an even number, we can be pretty assured it is a eukaryotic cell. And as I mentioned, the species with 8 chromosomes in each cell is the fruit fly. Here we have a diagram of DNA's structure. We looked at this before. DNA's structure can be described in a couple of ways. We can describe its individual parts, we can describe its shape when it is unwound, and we can describe its shape as it would exist in the cell as a double helix. So let's get started. This circle surrounds the repeating subunit that makes up DNA. That repeating subunit is called a nucleotide. So grab the nucleotide label and pull it across. Now that nucleotide, which repeats here 8 different times, has 3 parts in it. The circle part is the phosphate group. The pentagon, the little house structure, is the sticky sugar that connects both to the phosphate and to the third structure inside. That is the sugar, and in DNA, that sugar is deoxyribose. And the rectangular structure, the third part of the nucleotide that comes out into the middle of DNA, is the nitrogen base. Now you can see there are two sides to DNA, a chain of nucleotides on the left, a chain of nucleotides on the right, and they meet at their bases. These dotted lines show that it is hydrogen bonds holding together the bases, and therefore holding together the two sides of DNA. But on the sides, we have solid lines because it's a stronger bond. In fact, it's a covalent bond that holds together the sticky sugar and the phosphate group in a single nucleotide and connects that phosphate to the sticky sugar of the next nucleotide. When you look at DNA unwound, not as its individual parts, but as a whole, it looks like a ladder that you could just climb up the rungs, the steps. The back of each is called the backbone, and that's where the sugar and phosphate repeat over and over. The bases form the steps or rungs of the ladder. DNA in a cell, though, winds up into this double helix shape. What does the A stand for that we see in these first two ways of showing DNA structure? If you look at the key, you'll see that A stands for adenine. What about the T? Look at the key again. The T stands for thymine. And remember, when I say A and when I say T, what I'm really talking about are nitrogen bases that would be found joined together in the middle to form the steps of the DNA ladder. What does the G stand for? Well, here in the key, we see the G stands for guanine. And the C stands for cytosine. You may have noticed every time we have an A on one side, it is paired with a T on the other. You see that again down here. You see that pattern repeating in the DNA ladder. The base letter A always goes with T. Adenine always pairs with thymine. What about G? What does it always pair with? G always pairs with the base C. And C stands for cytosine. Next question, how many sides does DNA have? It has a left side and a right side. So that is 2. And the DNA ladder is twisted in the nucleus into a double helix. Just as a reminder, the pattern that we just saw is called base pairing. A always pairs with T, C always pairs with G. This is called complementary base pairing because A is the complement or the matching piece to T, and C is the complement or matching piece to G. One easy way to remember that is apples in the tree, A pairs with T, car in the garage, C pairs with G. Let's try it out. If there is an A on the left, what will be on the right? Well, apples in the tree, A pairs with T. If there's a G on the left side, what will be on the right? Well, car in the garage. What pairs with G? C pairs with G. Here we have a T. What will pair with a T? An A. And here we have a C. What will pair with a C? A G. When you look at DNA in two different organisms of the same species, like here two humans, you may notice that the DNA has the same chemical structure, but the sequence may be different. So here's Jenny's DNA and Juan's DNA. How are they alike? Well, both of their DNA are made of nucleotides. Both have a ladder shape when unwound. Both have the same complementary base pair rules. A always pairs with T, and G always pairs with C. But in Jenny's DNA, we have on the left A, G, T, A, C, G. That's the sequence of the bases on the left. In Juan's, we have G, T, T, C, C, A. So the sequence of the base pairs, even though they have the same number of them, is different. DNA is not the only nucleic acid, and so it's worth mentioning here because its cousin, RNA, will become very important later, the similarities and the differences. Both DNA and RNA, its cousin, are nucleic acids, which means they are made of nucleotides. And they both have nucleotides with 3 parts. Each nucleotide has a sugar, a phosphate, and a base. But there are lots of differences. Watch this video as your last action on the presentation today. And as you watch the video, fill out this chart. Then come back here and check your answers. Did you watch the video and fill out the chart? If so, check your answers here. You can confirm them with the diagram. DNA has 2 sides. It's double-stranded. RNA has just one. It is single-stranded. DNA uses thymine as one of its 4 bases, but RNA replaces the thymine with uracil. And the reason for that is its sugar. DNA's nucleotides use the sugar deoxyribose, giving it its name, deoxyribonucleic acid, but RNA uses as its sugar ribose, giving it its name, ribonucleic acid. I hope this helps. We'll see you again next time.